I am delighted to introduce our speaker today, Barbara Engelhardt. Barbara was born at Columbia Presbyterian Hospital on the Upper West Side.
Yes.
There's a point to this, Ed. All right? Yeah. On the Upper West Side of Manhattan. And in fact, her father was working at Columbia Medical School at the time, where he was a geneticist. He had studied with Norton Zinder, working on things including nonsense-mediated decay. And so somehow in Barbara's very early start in this world, she had a connection to genetics. But it took her a while to remake that connection with genetics. And the peregrinations were something like this. As a student in high school, she loved pure mathematics, the beauty of pure mathematics, this intellectual thrill. She went to college at Stanford University, where she was put off a little bit by the entirely pure theoretical nature of what was done there, and to some extent the notion that the departments were filtering people out, and got captured instead by the excitement of statistics and computer science to bring the love she had of mathematics into those kinds of interesting connections to the real world. And she worked with Daphne Koller, where she did a master's at Stanford, and then went to do a PhD at Berkeley, where she worked with the superstar Michael Jordan. There is another superstar, Michael Jordan, but in the computer science world, this is the relevant superstar. And as compared to the other one who does sports, he is still active. And one can have longer careers and fewer injuries in computer science. And with Michael Jordan, he, as he does with all of his students, said, we don't do things in a vacuum. If you're going to work on computer science, you need to be connected to some field, some problem, something that's providing the driving inspiration. And it was natural for Barbara to make that genetics, because of a whole bunch of people around Berkeley who she knew who were doing genetics, because of this early history, et cetera. And when she finally got her PhD, she went on, not to do a postdoc immediately, but to spend a year working at a small startup that had just been organized. She was employee number 13 at 23andMe, where, well, you can get her privately to tell you over a drink what that was like in the earliest days of 23andMe. Then went on to do a postdoc in Chicago in this incredibly rich community of human geneticists there, working with Matthew Stevens and many of the other people, Jonathan Pritchard, Carol Ober in that orbit. Took up a faculty position at Duke and a faculty position at Princeton, where she is today an assistant professor in the Department of Computer Science and the Center for Statistics and Machine Learning. So she's a card-carrying machine learner who is also deeply steeped in human genetics and is interested in the questions of how to build high-powered models, statistical models for understanding the real mechanisms of human disease. And she's going to talk to us this morning on structured factor models to find interpretable signals in genomic data. Welcome.
Thank you, Eric. Okay, thank you so much for that amazing introduction. Never had anything like that before. That was good. Also, thank you, Aviv, for the invitation. It's really been an amazing 2 days here, and I'm looking forward to another one after this. And it's great to talk to all of you about the incredible work that's going on here. Okay, so today I'm going to talk to you a little bit about some models, some statistical models that we've been developing to study genomic data. So the general theme of the models that I'm going to talk to you about today is that they are models for exploratory data analysis, or EDA. And the general idea behind these models is that you get some observation. Oh, I don't know if this is going to work. Can you guys see that? Sort of. You have some observation of genetic data, genomic data of some sort, and you'd really like to be able to visualize it in some low-dimensional setting. You'd like to be able to explore it and find patterns. And the idea is that when you find these patterns, when you find these trends among the samples or the features in these data, then you can develop these hypotheses. And we're going to talk a little bit about what some of those hypotheses might be. And then you can test these hypotheses in another data set, do experimental testing. The focus of exploratory data analysis is really thinking about how to interpret this fitted model. And again, we're going to talk a lot about that in this talk, rather than prediction. So I'm not necessarily interested in thinking about someone's risk. I'm thinking about understanding what components of a genome, for example, lead to that risk. What are the mechanisms of that risk? And finally, when you think about exploratory data analysis, it often involves a visual analysis. So often principal components are used, and we're going to look at a few examples of sort of visually how we can identify these patterns in a low dimensional setting. So we can contrast this with sort of the bread and butter of statistics for the past 150 years, which is hypothesis testing. So when we think about what we normally do when we have a specific hypothesis, when we know a pattern that we're going to look for in data, again, we have this observation of data. We come in with our specific hypothesis. Maybe we've actually gathered the data to specifically support a test of that hypothesis. We run it through our hypothesis test. We get a p-value. And then we can publish that, of course, if the p-value is significant. The thing that's important here, though, is a very careful definition of the null. In other words, what would these data look like if this hypothesis were not true? Okay? And what we're going to notice is that, in particular, when we think about exploratory data analysis, there's no such thing as a null. We don't really understand this idea of what the data would look like without this particular pattern being true in the data. So one argument that I'm making, and I think a lot of the work that I do hopefully will support this in this talk, is that exploratory data analysis is undervalued in applied statistics and genomics because it's really difficult, because it's a very, very difficult thing to do. And even more than that, it's very, very difficult to validate and reproduce. So the idea is that many of the assumptions of these models that we use to, for example, think about dimension reduction are incredibly poorly characterized, whether it's assuming normality, non-heavy tails, non-stationarity. All of these assumptions need to be very, very clearly spelled out in order to be able to essentially condition the patterns that we find, the hypotheses that we come up with on these really important and critical assumptions. There's no null hypothesis. Like I said, I think this is also critical. We have no idea what these data would look like, really, if a particular pattern that we were interested in didn't exist in them. There's no test of statistical significance. I can take random data, project it down, and I will get some kind of pattern, but I need to figure out what the statistical significance of that pattern is. I can't do that without a null hypothesis. Often the conclusion that we look at, that we get from exploratory data analysis is going to be based on qualitative evidence, visual representations, for example. And also the results in exploratory data analysis are often hard to validate and to replicate. And part of, I think, the reason that my work has been fairly well received recently is because we're working really hard on sort of creative and interesting ways to validate the hypotheses that we come up with in these exploratory data analysis. Okay, so let me give you some examples from my work and others of exploratory data analysis for genomic data. So these two plots are both from this paper from Faculty of 1000 from 2015. And this was a reanalysis of this data that involved, let's see, it's mice and human. And we have a bunch of different tissues from both mice and human. And the question was, this is from Mike Snyder paper, 2013, I believe, PNAS. And in that paper, they did a dimension reduction. They used 3 principal components and showed using that analysis that, in general, mouse tissues clustered with mouse tissues and human tissues clustered with human tissues, as opposed to mouse and human livers clustering together, et cetera. This reanalysis actually did a very similar thing, where in this case it used 2 principal components, but it controlled for batch. And it turns out actually that in the data that they were looking at, mouse and human were actually run, the RNA sequencing was run in 2 separate batches effectively. So controlling for batch basically completely destroyed this signal of mouse and human clustering together. And in fact now we see that actually, sort of a lot of the tissues, if you look carefully, are going to cluster together much better than mouse and human. Similarly, we can look at this in terms of a heat map, and again, this is a visual representation, but looking at this dendrogram over here, you can see again that the tissues are going to tend to cluster more tightly in the dendrogram than the species themselves. And really, the point of this analysis is that not that the original paper was wrong, but that there was not enough signal to support either hypothesis, right? That you could look at it either way. And both would essentially, you'd find signal both ways. So this is part of the difficulty of exploratory data analysis. In other types of models, one thing that my group looked at was these destruct plots, which I just think are phenomenal. So what we're plotting here is for every single column, this is going to be a single individual. And the colors then represent ancestry assignments of a proportion of their genome to a particular ancestral population. So for example, this is going to be the PopRes data set, which is about 1,000 individuals from across Europe. And you can see that in terms of the inferred ancestral populations, pretty much everyone in Europe is some sort of admixture, right? You see a lot of admixture. No one's sort of purely any one type of European ancestry. Whereas if you look at these HapMap samples, HapMap 3 data, you can see that there are definitely some populations that are much less admixed according to these inferred ancestry populations. Okay, so just as a talk overview, we're gonna talk about factor analysis, which is sort of the general statistical framework I'm going to use for exploratory data analysis. And then I'm going to talk to you about 3 specific examples of exploratory data analysis that I've looked at in different genomic data sets. And the point of these 3 studies was, first of all, understanding the genetic basis of high-dimensional traits. We're thinking about gene expression traits here. Then thinking about differential and context-specific interactions. We're going to look at co-expression networks and how to build those. And then finally thinking about shared latent structure across 2 observation types. And the observation types I'm going to look at here are gene expression data and image data from the GTEx project. And then very briefly, I'm going to talk about some current work I'm doing that is not quite published yet. Okay, and feel free to interrupt me too. I tend to talk fairly fast, but I'd love to hear your questions if anything's vague. Okay, so we're going to start out thinking about Bayesian latent factor models. And the general framework is going to look like this for the entire talk. This is sort of the general model we're going to be looking at. So Y here is going to be our representation, in this case, of gene expression data, right? Where we have the number of genes on the rows and the samples on the columns, okay? We're going to decompose this matrix Y into 2 matrices that at this point have been unobserved, but we're going to try to do this mathematically, into a lambda matrix, a P by K matrix, and a K by N matrix X. And we're going to talk about lambda as the loadings, the factor loadings, and X as the set of factors. And then our epsilon here is just going to be white noise, and this is just going to be Gaussian, independent Gaussian white noise. And the idea here is that this high-dimensional matrix maybe can be decomposed into much lower dimensional matrices. So K here, which is the number of columns of lambda and the number of rows of X, may be very, very small with respect to the original values of P and N and Y, okay? So then when we think about a specific element, a specific observation, a single individual and a single gene in Y, this is just going to have a Gaussian distribution, which is going to be the sum over the relevant factors and factor loadings, and then a precision that is going to be gene specific. It's going to be J specific. So every gene has its own variance term. Okay, so this may seem fairly vague right now, but I'd like to tell you that a lot of the statistical methods that you probably use every day are actually examples of this with slightly different constraints. So for example, principal components analysis is exactly this model, except that we've required that the factors here, the factors here and the loadings here are going to be orthogonal and orthonormal respectively. Okay, so we have some constraints on our factors and loadings, but besides that, actually, principal components is just a maximum likelihood estimate of this model. Admixture models or latent Dirichlet allocation are actually can be similarly described. In this case, both the factors and the loadings are going to be on a simplex. In other words, for every row and column, the values are going to sum to 1 and be greater or equal to 0. We can think about this in the context of non-negative matrix factorization, which has gained a lot of press actually recently when thinking about RNA sequencing data and sequencing data generally, where this basically involves count data, where we think about sort of a Poisson residual. So we can actually work with count data now. And there's a lot of nice sparsity in these models as well. And then we have also the idea of factor regression models. This is related to linear mixed models, where we have these two terms that are unknown, but we may put a known covariate in there. So if we have batch or we have sex of the samples, then we can include that in there and jointly fit this fixed effect in this model and be able to control for that when we think about this low-dimensional linear projection. Okay, so there are a lot of difficulties just applying these models out of the box, and that's why we can't just go to Python and run factor analysis and hope to get a real answer. So in particular, when we think about this model, it's actually going to be invariant to orthogonal rotation of both of these matrices, scale of both of these matrices, and label switching. Okay, so in other words, I can do orthogonal rotations of lambda and x and still get the exact same answer. So that makes me very nervous as someone who actually tries to interpret these values. The invariances actually mean that interpretability is incredibly difficult. So first of all, the factors in a standard factor analysis, not PCA, are not necessarily going to be orthogonal. They may be well correlated. And actually, we can talk offline about this, but often the correlation is going to be essential to interpretability. And we'll see that actually as something that comes up later. The factors will not be comparable within or across applications because of this scale invariance problem. Across applications is actually also because of rotation. I can run this 100 times on a data set, and I will actually get 100 different rotations where I can't really match the factors. As I said at the beginning, there's no approach to quantify how likely it is to see one of these factors versus a null, right? We can make up a lot of signal, and the proportion of variance explained of each of these factors may be very, very small. So the bottom line, I think, is that we must be incredibly careful when we use these factors outside of the sort of the standard data exploration questions. So in particular, I've seen a lot of these very high-dimensional data sets, for example, studying cardiovascular disease, where they project these high-dimensional phenotypes down to a low-dimensional basis, and then they just use the factors that they've estimated to do genome-wide association scans. And this makes me incredibly nervous because it's impossible to understand, to interpret exactly what phenotype we're mapping. And furthermore, it's also impossible to reproduce those exact same factors across different data sets. So I would caution against doing that sort of thing. OK, so how can we build a better model? Because we'd like to be able to do this in a reliable way. So first of all, we think about these models being robust to sample bias. And without being too much of a downer, I'm going to tell you that I use model-based methods because they are inherently robust to sample bias, unlike something like PCA. So PCA, for example, if you think about gathering 10 times the number of samples in a particular sort of region of this space, then the proportion of variance that needs to be explained with those 10 times larger samples is going to grow substantially. Okay? And so that's why when we do PCA and change the composition, the bias, the samples, we can have wildly different principal components. Whereas when we think about models like factor analysis that really model covariation as opposed to variation specifically, we are actually quite robust to sample bias. We also want rotational non-invariance. When we throw these models at a data set 1,000 times, we would like to get approximately the same set of factors every single time, the same basis, in a rotational non-invariant way. We would also like non-orthogonal factors. It often is the case, as I'm sure many of you have seen in your own work, that it turns out that the basis of a particular mechanism is actually shared across a number of mechanisms. And we would like to get those correlated mechanisms as separate factors. Okay? So sometimes correlation is actually OK when we think about pulling these factors. Interpretability. We would like to go back and understand actually what elements of the features and the population are leading to these factors, to this signal that we're pulling out of the data. Computational tractability is obviously essential. Given the types of data sets that we're looking at, that each of you have right now, computational tractability, we can't spend months fitting one of these models. And then finally, something that's a little bit more subtle, but again, we'll get into later, is we'd like to get the dimension from the data. So I told you that there was this latent dimension k. And that's arbitrary. We'd like to not run every k when we fit these models. We'd like to let the data show us what k is. Even when we have all these behaviors, I'm going to, again, caution you to consider the invariances in the models that we're applying to these data when actually trying to interpret the factors in a good way. And in particular, I would like to motivate driving the validation, the sort of creative validation of the factors. So one thing I feel like I always have to explain is why I'm a Bayesian, besides my history that you've heard. This is something that goes on when we, get feedback from NIH proposals. But in general, there's a real trade-off, I think, in statistical modeling between expressiveness and computational tractability, right? On one hand, we would like to model as much complexity in the data as we can possibly. But on the other hand, we need to make sure that these models fit the data in a reasonable time frame. And in general, when I build models, and I should say I'm a very practical person, so I'm not going to do Bayesian for the sake of Bayesian. When we think about genomics and neuroscience data, they're often actually not really this sort of canonical big data at this point. Although, with due respect to everyone in this room, you guys are producing the highest dimensional and most well-powered. Your sample sizes are just incredible. It's really phenomenal. But often in my world, where I don't have access to n equals 100,000, these aren't very big data sets. So they're sample limited. In other words, it costs a lot to sequence a particular individual, but when we get a sequence from an individual, we have 4 million SNPs. They're very feature rich, a lot of correlation among these features. But in general, actually, a lot of the statistical models and statistical theory that has been developed don't support these kind of data. And I think there's a lot of room to grow statistically in terms of analyzing these feature rich but sample limited data sets. So what I'd like to argue is that actually highly structured models are essential to being able to recover the interesting biological signal, the mechanisms that we can find in the data we're talking about. And I will show you some examples of how additional structure on models actually did save the day in a couple places. And most importantly, I think, actually, when we think about the models, we would like to be able to interpret the estimated parameters in terms of a mechanistic interpretation. The bottom line about all of the work that we're doing is to really understand how genotypes lead to these complex traits. And unless we have a model that allows us to sort of open the box and explore what's going on in our data, I don't think they're worth that much. So these are why I choose these type of models. So that said, I think there are actually many different ways to, with these complex structured models, achieve computational tractability. And so all of the models that I'm telling you today have been fitted using variational inference and variational EM. We use stochastic variational inference in a few newer papers, which works phenomenally well. Elsewhere, I have papers on using integrated nested Laplace approximations to approximate integrals very fast. And actually, we have a new paper out, I guess it's going to be out soon in JASA, about using generalized method of moments for these admixture models for population structure that has some really nice properties. So I think there's ways to get around, or not get around, but achieve computational tractability. Okay, so let's actually jump into some of the specific models. So here again, as I said before, Y is going to be a matrix of P gene expression levels from N samples. And the latent factors here are going to represent many components of gene expression variation. So these are going to be all the things in the gene expression data that have led to variation across these samples. And so in particular, we can pull out factors that approximately represent batch and library prep, and I'll show you what that means. We can think about biological covariates, so tissue heterogeneity, sex, age, BMI of the samples. Sample ancestry, population structure actually plays a large role in differential gene expression levels. And then, importantly, but very sparsely, genomic covariants play a big role here. So when we think about single SNPs in subsets where specific genotypes occur in subsets of these samples, we can pull out genes that are up or down regulated based on those SNPs. So we're going to look at that a little bit. So this is how I sort of motivate these very, very sparse factors. So sparsity basically means that in our lambda matrix, this is our loading matrix, we're going to have a lot of zeros. So anywhere that we have a black mark here, a black element here, it's going to be a non-zero value. So I'm expecting a huge amount of sparsity in lambda. And so, for example, if I think about a single factor loading, this has 3 genes in it, where these 3 genes are represented, for example, by a cluster in a network, in a co-regulation network, where perhaps there's a single SNP somewhere here that is co-regulating the expression levels of these 3 genes. The catch about having this much sparsity in one of these low dimensional projections is that they no longer are low dimensional. In order to capture all of the different subgroups, the cliques in this network, we actually might need a very, very large number of factors. In other words, our k, our dimension that we're reducing this space to, actually might be gigantic. So this is called an overcomplete basis, where we actually have a basis that is actually larger than the original m and p. Okay, so the way that we actually practically add sparsity to this factor analysis model, and right now we're only adding sparsity to the lambda matrix here, the loading matrix, is using, so first our model is exactly as we had it before, where it's going to be the linear combination of the loadings and the factor values and some Gaussian noise. And we're just going to put a very, very simple prior on x, our factors in this case, which is just a normal 0, 1 distribution. So in order to regularize lambda, what we do is we actually have 3 layers of regularization. So this TPB is the 3-parameter beta distribution, and I'm going to show you that in the next slide. And we actually have a global, we regularize globally. We shrink everything as much as possible in this entire matrix. We allow each factor to have its own regularization, so we keep shrinking each factor separately. And then within a factor, within one of these columns, we actually allow each element to also be shrunk individually. Sounds like overkill, but I'm going to explain why we do it. And then we draw each of the elements of lambda from this Gaussian distribution. So let's go into what that actually looks like. So this is the 3-parameter beta distribution, and you don't need to parse it too much, except to say that it looks like a standard beta distribution, except that there's an additional term here, psi, which is used to modulate the variance term. Okay? So if you think about what this looks like, when we set, for example, when we set alpha and beta both to be 0.5, this is actually what's called a horseshoe distribution. Okay? It looks very much like a horseshoe in that it's high at 0 and it's 1. So the reason this actually regularizes these variables, so we're thinking about, again, shrinking these lambda variables as close to 0 as possible. So the way that it does this, again, is we have these lambda variables are going to be distributed normal with mean 0. Okay, so we have a Gaussian distribution with mean 0. And the variance is going to be this 1 over psi term, where the psi term is going to be specific to that exact element of the matrix, minus 1. And so when we pull these psi values from or the phi value, sorry, from this 3-parameter beta, when they're very, very small, this variance component is gigantic, okay? So if you think about what that means, it's going to be a very, very, very flat distribution across the reals. And so this value is allowed to essentially be whatever it wants to be. There's not going to be very much constraint to be small. But when this value is very large, then this variance is effectively 0, okay? And so what that means is that we have a 0 variance spike at 0, which is essentially a point mass at 0, which forces this lambda variable to be 0. Okay? So we essentially have this sort of 2 components where either this is very flat and essentially imposes no regularization on these lambda terms, or it's very, very spiked at 0, and you're essentially forcing them to be 0. This has some incredibly strong regularization abilities. In other words, things close to 0 are very, very much shoved to 0, whereas things outside of this range are left alone, not regularized. So why 3 layers? So the global layer actually basically tries to shrink everything as much to 0 as possible. So we take these components of lambda, and basically everything in this matrix is as close to 0 as possible. When we look at the factor-specific regularization, so each parameter for each of the different factors, we actually rescue a few of these factors. So the ones that are supported in the data will not go away. They will be there still. And this rescuing actually gives us this non-parametric property, effectively, where the number k, the dimensions of our data are actually driven, or the dimensions of our factorization are driven by the data. And then finally, the local layer that we have here, the local shrinkage allows us to have element-wise sparsity. So within one of these columns, we can have sparse elements. So we played around with this model for quite a while, and it simply didn't work on gene expression data. And the problem here really, as pointed out by Leak and Story 2007, and actually the paper that really turned us around on this was a paper coming out of Alan Wilsky's group in 2009, where they basically described data, and gene expression data is one of these data types, that is not only sparse, but also has a low-dimensional representation. So in other words, not all the contributors to gene expression variation are going to be sparse. Not all of them are going to be driven by SNPs that are eQTLs that are regulating a small number of genes. Sometimes batch effects and population effects are going to regulate a huge number of genes, and we need to control for those explicitly. So what we did here was we basically just added a two-component mixture to these local regularization parameters, which allowed either the local regularization with this 3-parameter beta, or just pulled the regularization parameter from the factor regularization, which basically means that you're going to have a dense factor there, okay? So it's going to have non-zero values everywhere. We just put a very simple beta Bernoulli distribution on top of these variables pi, and we found actually that fitting these models, it was very robust to our values of pi. Okay, so we applied this model, we'll call it SFA mix, to gene expression data. And the first thing I wanted to tell you about was when we think about the dense factors. So these are going to be factors where there's basically no zeros in the loadings. And what we see is if we look over the 32 factors we found from this data set of about 480 individuals, we can look at the correlation of these factors with things like exposure batch or age or BMI or smoking status. And we see that there is a huge amount of correlation. So in particular, some of these factors are very well correlated with batch, with gender. Age is a little bit over here too. But one thing I want to kind of impress on you about this is that no single factor accounts for all of batch, okay? Because of this rotational invariance problem. So again, if I ran this 100 times, you would see 100 different matrices where batch effects were essentially explained and controlled for in different factors, okay? So this is where the interpretation of these factors, I think, gets a little bit difficult. But still, we can see that, of course, our dense factors are going to control for the things that we know have ubiquitous effects on variants of gene expression. So the next thing we did was think about how to validate some of these sparse factors. And so we did this using the idea of expression QTLs. So an expression QTL, very simply, is going to be a genotype, in this case, a genotype from a set of individuals. And here I'm using box plots. Gene expression levels are going to be, of a particular gene, are going to be on the y-axis. And what we're doing is basically plotting the gene expression levels for each of the individuals for each of these 3 genotypes. And in this case, we see that the means are approximately flat. In other words, the means are approximately the same across the 3 genotypes, and this is not a signal of a genotype driving gene expression levels. But here, we actually see a nice slope when we think about linear regression between the means of these 3 genotypes in terms of their gene expression levels. And we can argue that this is actually quite a strong eQTL, expression quantitative trait locus. So we did something slightly tricky, which I warned you not to do, but we did it anyway, which is we took a factor, each of the factors, in fact, that had a sparse loading, and we just used this to do association mapping, okay? So we used this instead of the gene expression levels to describe the individuals across our data set, and we used this as the response in a linear model. And so with this, again, now this is going to be one of these factors is going to be the y-axis of this association. And then we do this for all genes that are local, all SNPs that are local to the genes that are represented in the sparse, non-zero elements of the factor loading. So then when we do this, we can actually see a number of really cool things. So first of all, what this plot is showing you is this is the log-based factor, which is essentially the negative, not essentially, but you can think of it in terms of the scale of the log 10 p-value, negative log 10 p-value. And this is for the factors that we had. Whereas this is going to be the log 10 base factor for just the univariate test, where we're just testing all of the genes against all of the local SNPs. And what you can see here is that this is a 0-1 line here. We can see that we have substantially more statistical power to identify eQTLs. And the red dots here are going to be the cis eQTLs, the local ones, and the sort of green ones are going to be trans eQTLs, where they're actually off chromosome in this case. And again, we're not just finding cis eQTLs over here where we see really strong signal. We're actually enriched also for our trans eQTL signals here, which is something really nice. And the argument that I really want to make about this is not only are we finding interpretable signal, we actually are finding factors that represent sets of genes that are co-regulated by particular SNPs. But the benefits of the structured model are substantial. So we tried this for the only other sparse factor analysis method that ran on data this large, which was SPC, sparse PCA, from Daniela Witten in 2009. And running it, we found 5 times as many EQTLs doing our approach as opposed to theirs. And I think it's, again, because of the very flexible sparsity-inducing structure that we had, where it really allows a huge diversity in terms of the number of genes represented in each of these factors. You have a question?
Sparsity saves you from the variance a bit too, right? If you rotate, you're not sparsity.
Exactly, that's exactly right. So if you have an underlying set of patterns that's sparse, we can run this 1,000 times, and we're actually going to find almost identical factors based on the sparsity. That's exactly right. So that helps you with the rotational invariance. Yep, exactly. Another question? Yeah?
Yeah, maybe I missed it, but you said before that this is something that you don't think is legitimate, and you did it here and you're showing.
Oh, sorry. I shouldn't have said I don't think it's legitimate. So, well, let me say that properly. So, so the reason that we can do it here, let's just talk about this. So the reason we can do it here is because, first of all, we have this rotational invariance that you just mentioned, Alex. And second of all, because we're actually validating it with the univariate analysis, right? So when I plot this point, I'm basically saying that all of these genes versus all of these SNPs actually do have, with FDR 5%, significant association with the genes that we find associated with them. So I'm actually validating with the univariate data set. We're just powered up to actually make these calls. Yep, good question. Okay. So, so the next part of the talk, I just want to make sure we're doing okay. Yep, is on gene co-expression networks. So I think it's not very difficult to argue that gene co-expression networks have been incredibly useful in terms of sort of being important and usable latent structure in gene expression data. So in particular, thinking about regulatory relationships among genes, thinking about studying the co-regulation of gene modules as opposed to single genes. Thinking about how gene modules will change for particular perturbations, and particularly thinking about drug targets as well, looking at the important nodes in these networks. There's another type of network that I think is very interesting, which is differential gene co-expression networks. So this particular network, it's kind of hard to see, but it's going to be a network where the genes in this network are actually differentially co-regulated among males and females. And often this is characterized, if we look among the set of genes in this network, as actual differential expression among males and females, where you can just sort of eyeball this plot and see that they are differentially expressed. So this is nice because we can compare gene modules across different conditions. And I can actually find, again, gene modules that are very predictive of pathological conditions as opposed to normal, for example. So one type of network, that co-expression network, that hasn't really been discussed before, although it's been attempted in a few cases, are these condition or context-specific gene co-expression networks. So here's a network where the relationship between each of these genes is actually going to be only identified in females. Okay, we're going to describe a relationship. Each of these edges is going to be defined by partial correlation. And the nodes are going to be gene expression levels. And here, in this case, we're only seeing these edges in females. We're not seeing the edges in males. And similarly for this network that we only find in males. So context-specific gene co-expression networks are going to capture these condition-specific interactions, these interactions that really only occur in a single condition. You can think about conditions as anything. They can be sex, tissue, exposure, disease status, et cetera, anything you want. And I would argue that these networks are uniquely informative, and you're going to see this a little bit in the results, I think, that really we can understand interactions that occur very specifically in a condition that really don't have parallels outside of that condition. And thinking about targeting specific interactions that only occur in a single condition, and thinking about actually condition-specific regulatory complexes. So these groups of genes that really only interact together in a single condition. So the way we do this, and I talk too long, so I'm going to go fairly fast through this, but it's actually quite simple. So we have the exact same setup as before, except what we're going to do is we're going to put this same sparsity-inducing prior on X, on our factors. And so besides that, it's the exact same model that we had before, where we have sparsity-inducing prior on X, lambda and X this time. So the way, this is called Bayesian by clustering, and the reason it's called Bayesian by clustering is because if you look at a specific cluster here, a specific factor loading and factor, you can see that there are going to be a lot of genes in the factor loading and a lot of samples in the factor that are 0. right? That value is 0. The idea is that you're going to cluster, this is a clustering of both the genes and the samples simultaneously. So to see this a slightly different way, if we take the outer product of each of these k factors, you see these interesting matrices where you explain variation in a subset of the samples and a subset of the genes, but everything else in this particular factor is going to be blank. In other words, this covariation among these genes only exists in a subset of the samples. Okay? So this is by clustering. And again, as before, we're going to allow dense factors in both the factors and the loadings. Okay, so what we do with this. So the object is to actually understand the relationship among all genes. This is actually an incredibly difficult thing to do. We have P genes, maybe 20,000 genes, and only about 400, 500 samples on average for the data sets we're looking at. And so we need to be able to develop this covariance matrix of Y, where the covariance is, again, over these P genes, using some kind of regularized representation of covariance that allows us to invert this matrix. Okay, inversion is going to turn out to be important. So the way that we do this is actually we integrate out, in this factor analysis model, we integrate out X, and we get something that looks like this, where if we take lambda, and we take lambda transpose and multiply them together, and we add on our residual variance term, which is going to be a diagonal matrix, we end up getting an estimate of a low-dimensional estimate of the covariance matrix of Y. Okay, and this is beautiful because it's mostly non-singular in the sense that we can invert it because we've added this pretty strong diagonal to it, making it full rank. So what we do is we take this estimated covariance matrix, we invert it explicitly, we get what's called the precision matrix psi. This turns out, when we normalize it, to be just the partial correlation of these genes. So partial correlation, as I said, is sort of the semantics of these co-expression networks. These are going to be Gaussian graphical models or Gaussian fields or Gaussian Markov random fields. So you can call it a number of different things, and it's called different things in different literature. But the point is that each of those edges, again, represent the correlation between two genes A and B, conditioning on the values of all other genes. So in other words, when we see a partial correlation that we can infer is non-zero, that means that even with all the other genes that may or may not be co-regulating those two genes we're looking at, they have a direct relationship. So we're going to put an edge between the two of those. So we basically do that by thresholding the psi matrix using FDR, false discovery rate type approaches. We get a matrix that turns out to be a bunch of zeros and ones now, a symmetric matrix. And then we're going to take the genes, all of the genes on the rows and columns of this 0-1 matrix, and we're going to put edges everywhere that there's a 1 between a pair of genes. And we can get one of these networks. And I should say, when I show you these networks, the size and the color of the nodes correspond to something called betweenness centrality, which basically says for any node, how many times do the shortest paths between any other pair of nodes in the network go through that node? That's going to be betweenness centrality. Yep, so that's how we do that. So I should say that one thing that we do now is think about these condition-specific networks. So this is how you just get a network from the entire data set. But what happens if I want to find a network for only liver tissues, for example? So the way we do that is we actually build this correlation matrix now, or covariance matrix, based on a subset of the factor loading. So as opposed to include the entire factor loading matrix, we're only going to include those factor loadings where the corresponding factors have non-zero values in that context. So in other words, these factor loadings are the only factors that have non-zero loadings in liver. And so in samples from liver, and so we're going to pick out those and build this covariance matrix. So we're going to do the exact same thing then, which is given this estimate of the covariance matrix that's specific to liver, we're going to invert that matrix, get a precision matrix, do this thresholding again, and now we have a liver-specific network. Okay? I should say that, again, I think these context-specific networks are really special in that we've removed all the sources of variation that that are shared between liver and anything else. There's really no parallel covariation outside of this particular context we're talking about, in liver, for example. And then when we think about these genes in this network, they're co-expressed uniquely in this context. This is a really interesting thing and that I don't think has been explored, and again, we'll see in results. So another thing you can do is find these differential networks. So the way we do that is essentially the exact same way we do this, except we look for factors now that have using Wilcoxon rank sum test have different factors for males and females. So you often see all positive factors for males, all negative factors for females for a particular factor. And in that case, we're going to reconstruct the covariance matrix using only those factors. Okay, so so I applied this to the GTEx data set. This is consortium V6 data. Thank you to the Broad for doing all these analyses. So right now we have about 552 individuals, a little over 7,000 samples, so approximately 18 samples per individual. I don't know if I need to explain this very much to all of you, but I will anyway. About 2/3 male and 1/3 female right now. And obviously the magic of this data set is that we have 44 different tissues here, right? From many different individuals. We have whole genome sequences now, and we can really get a lot of very interesting context-specific, tissue-specific biology and gene regulation from these data. So the first thing we did was just to see if it worked, basically, to try to find sex differential gene co-expression networks. And this is the network that we found. And I'm a statistician, but this was awesome to see this network come out the first time, because you don't have to be the best biologist in the world to see that all of these genes are incredibly involved in sex differential biology. So a lot of these are X or Y linked. And very involved in sex differentiation, and it was just very exciting. So we also just plotted this heat map of males versus females in our samples, and again saw that there's going to be a very differential expression of these genes within the two different contexts. So then we tried pulling our female-only gene co-expression network, and it gets really, really murky at this point. So this is the network that we found. There's sort of one canonical gene here, ESR1, the estrogen receptor, that is canonically differentially expressed among males and females. Besides that, there's actually very little known about most of these genes in the context of sex differentiation. So I sort of made up a few stories here based on PubMed search, but the reality is that these are very difficult to interpret. And again, I think it's difficult to interpret because there's not differential expression here. There's differential co-expression. In other words, only in females do we see a relationship between each of these pair of genes. And one thing I wanted to say too, I don't think it's in here, but one of these genes is actually fairly recently been involved, implicated in its involvement in multiple sclerosis, which is a sex-biased disease. It hits women more than men. And it'd be really interesting now to go back and think about the role of targeting these particular genes for these sex-biased diseases. Okay, so as I promised you before, what do we actually do to validate these networks? So the way that we did this, and GTEx is of course unique in this sense, we have eQTLs, these expression QTLs for every single one of the genes essentially in these networks in a tissue-specific way. So what we do is we take, for example, a cis eQTL for this gene here, and we just test for relationship, for a eQTL relationship with only the neighbors, and we do that test only in this cell type. Okay, so this is a cell type specific network. If you think about this, this is actually going to be a test for trans eQTLs, these distal eQTLs, because often these genes are not linked with genes that are close by them on the genome. In fact, this is most of the case. And if you also think about it, when we actually run trans eQTL mapping for GTEx across the 44 tissues, we end up doing about, I'm forgetting the number, but it's going to be on the order of 12 trillion statistical tests, okay? So when we filter those tests based on the network, we effectively save 10 orders of magnitude of the number of tests, okay? But the shocking thing is that, again, using the exact same FDR as we do for the whole genome scan, because we have this very concrete hypothesis, we actually find approximately the same number of trans eQTLs using 10-fold fewer tests as we do in the entire data set. So this was kind of a phenomenal result. I'm not saying we get all of the trans eQTLs. On the contrary, we've removed most of the tests. But the ones that we do get, we can now go back and validate. And we're actually talking to some collaborators about validating some of these trans eQTLs in specific tissues. Okay, so time check. Okay, we're doing okay. Very quickly, I'm going to tell you about another model which basically combines two different types of observations. So we have Y1 and Y2 now. Y1 is gene expression data again. Y2 is going to be something slightly different. And so the idea is then that each of these different observations, so this might be genotype, it might be images, we're going to project this down to the exact same X space. Okay, so these factors are going to be identical across the two observations, but the mapping is going to be different. And the idea behind canonical correlation analysis is that we are finding this space X, this latent space X, that maximizes the correlation between these two types of observations. So what does that get us? I'm going to skip that. So if we do this with genotype and gene expression data, we find that when we look at a single factor in the canonical correlation analysis, we can do association mapping between each of the SNPs that are non-zero and each of the genes. And we find, in this case, significantly enriched low p-values for these associations, meaning, again, that they're going to be SNPs that are actually driving the regulation of the genes that were found in that component. So this is something that I think is even more exciting. We took from TCGA some BRCA data sets and also some images from GTEx, because GTEx has pathology, these histological images. And we put these images all through an autoencoder. You can't just put pixels into canonical correlation analysis. So we put them through a convolutional autoencoder to get about 1,000 features from each of the samples. And then we ran canonical correlation analysis, and we found these sort of phenomenal things, which is, so what I'm showing you here is for this particular variable, this particular component of CCA, these were the images that were very high in that component, and these were the images that were very low in that component. And we found a set of genes associated with the differences in these types of images. And when we look at the top Go terms for these genes, we find, again, some phenomenal things where these genes are going to be, in general, involved in extracellular matrix development, in ion channel binding, and cell adhesion. And what we can do is actually start thinking about finding genes that are involved in the differentiation of figures in these automated phenotyping of these images using this type of method. So I should say, similarly with GTEx, we found some features that very clearly differentiated, for example, muscle cells from non-muscle cells. And we can go on each of these factors I could look at for hours. They're pretty awesome. OK, so that's factor analysis. Really briefly, I'm going to tell you about some of the other current work that I'm doing. I've been thinking a lot about time series data recently, and we have this amazing collaboration going on with the University of Pennsylvania people. They gave us about 270,000 EHR, electronic health records from patients in their hospital. We have time series data on all these patients, and what we do is build a multivariate Gaussian process that allows us to track monitor the underlying patient state. This is interesting because then we want to, for example, predict when a particular patient is going to have septic shock, for example. We're using reinforcement learning techniques to try and guide and advise doctors on when to pull someone from a ventilator in a safe way. There's a lot more to do here, and in particular, I'm really interested in combining this with genotype data from the patients to see if we can find, for example, drug responses that are specific to a genotype. Another set of work that I'm doing with Tim Reddy and Greg Crawford down at Duke is thinking about using time series data of gene expression to develop these directed models. So the particular goal of this project is to look at glucocorticoids, which is an anti-inflammatory drug in lung cells. And we're getting 12 different time points of gene expression data after applying glucocorticoids to lung cells. And the idea is that we'd really like to figure out how to engineer these cells to essentially completely knock down the metabolic response, which leads to type 2 diabetes in the worst case for people who take glucocorticoids, and completely separate that from the anti-inflammatory response, which is obviously what we'd like to keep. So in these directed networks, we're first of all developing methods to build them, and then we're figuring out how to do active learning, how to do experimental design to tell us which are the most likely nodes we can knock down, the genes we can knock down to essentially remove the metabolic side effects of these drugs. So finally, I'm thinking about genotype data that regulates not just the mean value of a trait, but the variance of that trait. And so here, for example, you can see this is called a heteroscedastic model, where each of these genotypes actually have different variances. So you can imagine a SNP in your genome, perhaps, that leads to a much higher variance in height in humans, as opposed to a very, very small window variance in height with the other genotype. And doing this actually ends up finding not what you'd expect. It ends up identifying often a lot of places that are epistatic in the genome, where actually the higher variance is actually just an epistatic effect, which was kind of nice. So in conclusion, my group has thought a lot about exploratory data analysis. It requires incredibly well-structured models to get the kind of signal you want. When you're able to interpret and validate these models appropriately, we found a number of different things that I think are exciting in their own right in terms of the mechanistic biology that was uncovered. So sources of gene covariation, these gene co-expression networks, and specifically the context-specific ones, expression QTLs, and also transient QTLs specifically, and then thinking about correlations across high-dimensional data types. So what do I want to do next? I think for these particular models, we need to allow them to scale. We need to robustify the results. We can talk about that offline. The thing that I'm super excited about right now is actually experimentally validating some of the results. So I'd really love to talk to experimental biologists here about how to do that appropriately. Thinking about how to incorporate the biological knowledge that we all know into some of the priors to really get more sophisticated structure. Evaluating the statistical significance of the patterns that we find, thinking about how to improve statistical power to find the signals that we're looking at. And then obviously there's so much more exciting, complex data that we can think about. Very high dimensional, very interesting phenotypes, whether it's fMRI scans, more imaging data, time series data that I'd really like to think about in response to changes in genotype. So really briefly, I'd like to thank all of my collaborators and my students for doing all of this incredible work and my funding. Thank you.
I don't know where the microphone went, but I don't need it. Great, thank you. I think many of us have been worried about the fact that we're generating so much data at the Broad and more broadly in the community, and have been agonizing over how in the world we're going to analyze it.
Yeah.
And I think it's really nice to it was really breathtaking to see this within a general framework. Certainly in our world in biology, folks are used to getting up and saying, I ran a PCA, or I did this, or did that, and seeing all of these analyses as special cases of a coherent framework is very satisfying. And it's a really wonderful thing for us to see. And I think for many people in the audience for whom many of the details might have gone over their head.
I was told to make it mathy.
Well, no, but that's just fine. I think the point is that point came through even without having to necessarily understand each of the details of any particular method. So recognizing that this is a mixed audience of biologists and mathematical computational types, I throw it open to questions.
Yeah.
So I'd be one of those people that didn't get some of the details, but was interested in your context-specific networks.
Yeah.
And so if I understood you correctly, you used the existing external labels to define those contexts.
That's a
Did
great
you also
question.
try to define the context based on the data, and did that matter?
Yeah, that's a great question. So we didn't, mostly because those contexts may be ill-defined. So it could be, not necessarily ill-defined, but batch effects. Population effects we can sort of think about more specifically, but really I don't want to pull out networks that are specific to a batch. And the good news is that ignoring those factors that are over a subset of the samples that I don't care about means that all of those batch effects, all of those co-regulated genes that are not, or that are found across liver samples and a particular batch will be removed because we're just not including those factors in the estimates of the covariance for liver. Does that make sense? So we're controlling for all of those, but you're absolutely right. We do this in a supervised way where we use the labels to figure out the context. Yes.
As long as the context is not confounded with the batch.
Well, yes, but that's experimental design. So if someone did that, then we're all hosed. Yeah.
Other questions? Yeah.
Hi, a great talk. So throughout the talk, it almost struck me that factor analysis seemed like it was capturing something akin to human intuition. You know, you're trying to find patterns in the data. So I was wondering, first of all, is that a reasonable way to think about what it's doing? Is it's trying to come up with hypotheses? It's like a computationally rigorous way of intuition. And then does that suggest that you could then use factor analysis as one step in sort of a broader machine learning AI type framework to really find signal in the data?
Absolutely. So, so it's interesting because it's specifically linear, right? All of these models are linear. There's no nonlinear projection of the data at all. And so, I mean, maybe I'm giving us too much credit, but I think actually, you know, a lot of the patterns that we often see in data are very nonlinear. And so this is sort of the first pass, the simplest version of what patterns we might find. And because of that, I absolutely agree with you. So the reason we're doing all of these things is to find patterns that we can go back and explicitly test in data, right? That's exactly what we're doing. We're developing hypothesis, and I should say, I didn't really touch on it here, but one of the sort of imperatives of this framework statistically is that we need to use samples that are not the samples that we develop these hypotheses on. So really, we need to pull samples that are out of sample, that are new samples, that specifically will ask the questions about the data that we've hypothesized from, for example, GTEx. And so I think, again, that's a huge way that statistics and biologists can collaborate here.
So I was just wondering, for your specificity, male versus female specificity networks, I was wondering whether you compared your network through Bayesian model selection, for instance, to ensure this new hypothesis somehow, to be sure that your female-only network is not like part of the data you have not explained because this signal you don't know about.
Yes.
You see what I mean?
Yeah, I absolutely know what you mean. So, so I'll tell you. So first of all, we did the validation of specific edges using EQTLs. That doesn't answer your question. That's only an edge-specific validation. I completely agree with you. So we thought about this for a really long time. And if you think about what it requires, it essentially requires us to, let's say, simulate a bunch of data where we can go from a gene expression network, even context-specific, to the gene expression data itself. And it actually turns out that while we can invert the matrix one way, we can't invert the matrix the other. So again, I can tell you we tried a lot of stuff in that direction to figure out sort of the statistical power and validate the edges. But you know, there are so many settings on how to sort of fake this reverse inverse that we didn't feel comfortable making any real claims about it. It's a hard problem, and I'd love to hear your thoughts on it, actually.
One thing that I may have missed is how do you pick your reduced rank? Do you just choose a bunch of Ks, evaluate a marginal likelihood for each of them, and pick the highest? Or do you use some non-parametric technique like an Indian buffet or Dirichlet process? So
what we do is we don't use a non-parametric model for or non-parametric prior for any of the models that I talked about today, although we have that in some newer work. What we're doing here is actually, like I said, because we have this three-layer sparsity-inducing hierarchy, we basically start with 3 times as many factors as we think we need. So K is going to be like, let's say, 5 times our sample size, right? And then when we fit the model, that gets reduced by a third approximately. And so we end up getting the K from the data. But yeah, we have sort of rules of thumb about how to do that properly, but that's how we do it.
Great. I think in the interest of time, I'm going to ask you one last question in passing, and then we'll let other people come up and talk to you, which is, you referred to nonlinear things. What do you think about that? What's in the back of your mind about how to deal with those things that are inherently nonlinear paths in space?
Yeah, it's a great question. So, so in factor analysis specifically, there are some Gaussian process versions of this dimension reduction. So a Gaussian process is basically an arbitrary mapping from any feature space to any result. It's arbitrary functions, a distribution over these arbitrary functions, which is beautiful, and it's entirely free, right? And it is all Gaussian, so Gaussians are very easy to work with mathematically. But the problem is it's so much freedom that these models kind of get swamped. So we've, again, applied these nonlinear models, factor analysis models, to genomic data, and really it's difficult to see. So what do I think the right thing to do is? I mean, I think I think, again, because we have these nice hierarchical frameworks, we can sort of build in some ideas of what we think these nonlinear functions may look like, whether it's logistic type of function, whether it's going to be some kind of spline, polynomial, whatever it is. We have some intuition, I think, when we think about these patterns, about what these functions may look like. And then in that sense, I think it'd be really nice to explicitly model those nonlinear relationships in these models. And that's straightforward to do with kernelization, for example.
Cool. It'd be interesting at some point to talk about,
Yes, I'd love
just
to.
the high-dimensional data, just without any model, produce manifolds in the
Yes.
data,
Yes.
and then you can do your work on those manifolds. You can
Absolutely.
unfold those manifolds and.
Absolutely.
Cool.
And think about those manifolds as the phenotypes, these complex phenotypes.
Yeah, and you could stretch them
Yeah,
out and,
stretch
and
them
work
out,
on
that's
them
right.
those. Well, cool. That was great. Thank you so much, Barbara, and thank you all for coming.